everyone, welcome to our model on Embryology Week 2. These are the set of events that occur within week 2 of gestation, with two spans from the 8th day to the 14th day. And from those particular period, we have events like implantation being completed. Implantation starts around week 1, but it's completed around week 2. Then we have the development of the embryoblast and the development of the trophoblast occurring within week 2. So by the end of week 2, we should have implantation of the blastocyst being completed. We should have the embryoblast maturing into what we call a bilaminar embryonic disc. And we should have the trophoblast interacting with the uterine tube to form a primitive utero-placenta circulation. These are all the events of second week of gestation, and we'll talk about each of them in much more details as we go along the video. Starting with the development of the embryoblast. Embryoblast is a part of the fetal structure gotten from the inner cell mass. Remember that the blastocyst is made up of an outer cell mass, an inner cell mass, and then a blastocell in the center. The embryoblast will be gotten from the inner cell mass, and during the second week, the embryoblast further develops by differentiating into two distinct cellular layers, which is what we call the bilaminar embryonic disc. These layers would be the ventral hypoblast layer, and then the dorsal epiblast layer. So we have an epiblast and a hypoblast layer formed around the second week. These two together gives the flat ovoid shape, which we call the bilaminar disc. The epiblast layer tends to be columnar in its configuration of cells, whereas the hypoblast layer tends to have cuboidal-like cells. So this is the image of the bilaminar disc down below. You can see the bilaminar disc being formed by two layers of tissue, one being the epiblast in blue and the other being the hypoblast in green. During the second week, further development of each of these layers occur. Initially, we start with just a flat cellular layer of the epiblast and hypoblast, but as the second week proceeds, first of all, the epiblast would undergo serious maturation. The cleft begins to develop and eventually coalesces to form what they call a cavity, an amniotic cavity. So we go from a flat-shaped epiblast layer, an epiblast layer that forms a circular structure with a space in between. So the initial flat aspect of the epiblast just remains as the epiblast, and the aspect that forms a roof would be known as the amnion. So it's still formed from the initial epiblast, flat epiblast surface, but in this case we have a circular structure being formed, and at the center of the circle is a space we know as the amniotic cavity. So this is the amniotic cavity, AC for short. So something very similar that happens to the epiblast also happens to the hypoblast layer. We start a single cellular layer or the hypoblast, and cells from this single layer would migrate downwards to form a circle around the initial flat surface. And then these cells that tend to migrate downwards are eventually going to form a new layer or called the exocynomic membrane. This layer is in direct contact with the cytotrophoblast, as you can see. If you notice from that yellowy set of cells here, you have cells that migrate out and the line the inner aspect of the cytotrophoblast. So cytotrophoblast image. So we say that the exocynomic membrane tends to line the inner aspect of the cytotrophoblast, and in between the and this is really happens to the embryoblast. So start off again with the single cellular layer, the inner cell mass becoming two bilaminar, the epiblast and the hypoblast. We have the epiblast initially being flat, but then eventually forming cells that migrate upward to encircle it. The cells that are found on that flat area are called the epiblast. The cells that tend to migrate upward to form the circle is known as the amnion, and then between the amnion and the epiblast, we have the amniotic cavity. So that's what occurs at the epiblast tends to mature. Then the hypoblast also has something similar into it. That before we have a single layer of hypoblast and cells migrated down, circulates the cell blast, and the exocellular membrane is known as the yolk sac. Let's talk about development of the trophoblast. The trophoblast tends to also form two distinct layers. This includes the cytotrophoblast and the sensitotrophoblast. And each of these layers would further develop as the trophoblast is embedded into the uterine cavity. So, as we have two layers of the embryoblast, the epiblast and the hypoblast, we also have two layers of the trophoblast, the cytotrophoblast and the sensitotrophoblast. Each of these is in their in terms of also their function. Starting off with the sensitive trophoblast, this is an outer multinucleated zone of trophoblast, whereby there is no mitosis occurring. So imagine um, the sensitive trophoblast as cells that have mashed together, that are combined together, which are not separated by membrane. But we have like multiple cells combined together that are cytoplasm already connected, and as a result, these cells are forming like a syncytium rather than having individual cells. Another important thing about is that each of these cells, because of the fact that they combine together already, they can no longer undergo mitosis. It is part of the sensory trophoblast is a part of the trophoblast that usually comes in direct contact with the uterus during implantation, and it is the since the trophoblast is usually involved in the process we call decidual reaction. So usually when implantation is occurring, as the since the trophoblast is embedded into the uterine wall, it tends to erode a number of blood vessels and glands. And this erosion of blood vessels and glands usually triggers a reaction we call decidual reaction within the uterus. So as the sensory trophoblast embeds in the uterine wall, it forms isolated cavities, what we call lacunae in the at the site of contact between the sensitive trophoblast and the uterine wall. This lacunae tends to become filled with a mixture of maternal blood, and this comes as a result of rupture of endometrial capillaries as the sensitive trophoblast enters into the wall and erodes the vessels. This is important because of the fact that as Cephalopolis embeds the wall and forms those lacunae, and those lacunae are filled with blood. This serves as a form of primordial uteroplacental circulation. It's like the blood that is found within the lacunae gets a lot of oxygen and a lot of glucose that will be transferred from the lacunae into the sensitive trophoblast, and from the sensitive trophoblast into the cytotrophoblast and the embryoblast. So this is the way in which the placenta, which is going to be formed by the trophoblast, and the uterine cavity along its blood vessels tend to communicate, and we have blood being transferred or constituent of the blood being transferred from the uterine cavity or uterine wall into the placenta, and then eventually to the fetal structure. Whereas which is cell, is a combination of cells that are mitotically active. They are cells that tend to encircle the rest of the embryo, they encircle the embryoblast, they also encircle the cytotrophoblast internally. Compared to the cytotrophoblast, they contain cells that are mitotically active and they are always undergoing cell division. Cytotrophoblast usually tend to undergo cell division and eventually migrate to become the sensitive trophoblast. So we get sensitive trophoblast from cytotrophoblast. In addition, the cytotrophoblast tend to produce local mounds called primary coronary periodic villi, and these mounds tend to bulge into the cytotrophoblast. I'll show you a picture of how that looks like. Embryo so far, from the epiblast to the hypoblast, and then the other structures that are surrounding it. So if you remember, we start with a circle surrounding it. cells gives rise to what we the hypoblast also does something similar. The hypoblast initially starts as a flat layer, then gives rise to cells that forms membrane circulate. This membrane is known as the extra cellular membrane, and then the space between the extra cellular membrane and the epithelium is known as the yolk sac. The embryoblast completely. Then we have the trophoblast that now surrounds the trophoblast, and that's a major part of the contact with the crime wall. Now, what we relic sacs begin to develop around the second week, and that how that then comes about is that we have cells from the epiblast migrating out of the epiblast and between the extra coelomic membrane, as between the yolk sac and the amnion, and the cytotrophoblast. So just imagine cells starting to form, migrate from this epiblast and starting to form between these two structures. So between the cytotrophoblast here and then between the amnion and the extracellular membrane here, which is the yolk sac here. So we have cells that will just fill the space as shown here. So the cells that fill the space as I'm drawing here would give rise to a new set of structure we know as the extra embryonic coelom. So extra embryonic coelom or extra embryonic membrane, rather membrane, not coelom. Extra embryonic membrane are cells that forms from the epithelium and yolk sac, and then you divide the membrane into the part that is in contact with the cytoplasm. So, and the space is known as extra embryonic coelom, and it divides the entire membrane into the extra embryonic somatic and the extra embryonic splanchnic. They are not cavity and yolk. Well, that's the embryonic plastic in general. But if we have those cells forming what we call the extra embryonic membrane, then we have the space forming within that extra embryonic membrane that divides the entire membrane into parts, an extra embryonic somatic system and an extra embryonic somatic method. Now, I want to note something as shown in the image. Consider the space that would describe that the extra embryonic coelom has further expanded in this particular image. You can see that is larger, is the entire space here. This space itself is known as the extra embryonic coelom. And what you notice is that it tends to separate the structures of the trophoblast from the structures of the embryoblast. So if you notice, there's a new set of combination of structures that are formed in the embryoblast and then in the trophoblast. For example, in the embryoblast, we have the amniotic cavity here combined together with the blast and hypoblast, and then the yolk sac here, then wrapped around by extra embryonic splanchnic mesoderm. So Combination of those three things, right? The amniotic cavity and the epiplasm, the yolks are formed by the hypoplast and the coelomic membrane. Then the extra-embryonic splash to them surrounding to give rise to the embryo. That's the new form of the embryo. Then those they are the trophoblast. You see that trophoblast also form the third form of combination. So we have the cytoplasm outdoors, yeah. Then cytoplasm down here. Then you see that the cytoplasm now is lined by structures, which is the extra-embryonic matrix. So you can see the extra-embryonic matrix, the cytoplasm, and the centroblasm form a new set of structures. This structure gives rise to what we call chorion. Chorion is the condition of the extra-embryonic matrix, the cytoplasm, and the centroblasm. And that space that is dividing them, space dividing chorion from the newly formed embryoblast known as the chorionic cavity. Now, this is the full formation of the formation of the chorionic cavity. And what you notice as well is that though these two structures, the newly formed embryoblast also called chorion, and the newly formed embryoblast contains those three structures I mentioned. Um, Though they are separated by the chorionic cavity, there's still a form of connection. The connection is by the presence of this structure we call the connecting stop. This is the connecting stop here in the image is connecting stop. That's the newly formed embryoblast and the newly formed trophy. Puts everything in full circle. So it's about development of the embryo from the third from the um, day down to the 14th day of the 13th day, shown in the image. That's week two. That's the formation of the bilaminar disc. The bilaminar disc is gotten from the embryoblast. That's in two layers called epiplasm. That is shown in the image here. See the epiplasm here in red, and then the hypoplast here in green. Then these two structures were developed by forming cellular structures above and beneath. So the epiplasm forms cellular structures called is the amniotic here. Forms the amniotic cavity would have a roof called amnion, and then the floor formed by the glass. Then the glass will also form encircling structures. The encircling structures are formed by the glass is known as the exocellular membrane or Hughes membrane, shown here. And then the entire space between the exocellular membrane and the hypoplast, so the hypoplast here um, in green is the exocellular membrane here. That space is known as the yolk. That's what was during the development of the biomass. Now, for the trophoblast, development also tends to ensue in two fashions. We have the trophoblast developing in two major structures. We have the cytoplasm circles the entire biomass. Number of cells that have nucleus are intact, genes are intact, they are continually dividing. Then we have the on the outer layer, the sensitive trophoblast is made up of large amount of cells that are multinucleated, don't have a they are naturally active. And what this sensitive trophoblast have direct that the uterine wall, they are involved in the invasion and of the entire embryo. So then to embed and erode blood vessels, as you can see, as they eroding the blood vessels, they form spaces in the uterine wall. These spaces before are known as tunae. As you can see, that blood blood filled spaces that filled with blood and tend to serve as a form of protective communication between the uterus and its circulation and the embryo. And then further development occurs in the sense of the chorion shown in the last image here, that the chorion or the chorion form of a chronic form, where we have cells hydrogen entering into this protoplast and the bioblast in general. That's those cells we call them they form a structures we call the extra embryonic membrane. This extra embryonic membrane on time form a space within it. That space divides to an extra embryonic on one side, an extra embryonic on the other side. At the end of it, we have this large space dividing the embryoblast. The embryoblast become newly formed. We have three major structures: the yolk sac, the cavity, and then structures that are forming bilaminar. And then surrounding that, we have the terrestrial embryonic plant. That's the newly formed chloroplast. But the newly formed trophoblast also formed by three major structures. They have the sensitive trophoblast, the primary villi, the trophoblast, and then the embryonic somatic. Then the space in this last is known as embryonic. At the end of the day, we have the structure that connects them newly formed cells to the through the by way of structure we know as that actually for us the event the second week of neurogenesis. Clinical significance. So it's very important that during the development of the embryo around this time, we start to see the production of a very important hormone known as human chorionic gonadotropin or HCG for short. This hormone is used by the sensitive and it tends to be used as a very important tool or a very important marker of pregnancy. So if you do a pregnancy test, what we need to do is test the amount of human chorionic gonadotropin present in the blood. And around the second week of development, the embryo or rather the within the embryo should already be using large amount of human gonadotropin that is using pregnancy. Our module on embryology